Hello, everyone, and welcome to today's webinar, Streamline Your Path from Immunodiagnostic Development to Commercialization. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Anthony Chiulli, Director, Global OEM and Licensing, Protein and Cell Analysis, Thermo Fisher Scientific. Anthony, you may now begin your presentation.
Good morning, and thank you for joining today's webinar. My name is Anthony Chiulli. I am a Director of Global OEM and Licensing within Thermo Fisher's Protein and Cell Analysis segment. I started my career in R&D, developing diagnostic immunoassays, and have been in the business for over 20 years, partnering with customers globally to optimize a wide range of Thermo Fisher products and capabilities as part of their diagnostic and life science product commercialization. So Thermo Fisher is comprised of many different divisions. What I've shown in this slide here highlights the 4 segments within our biosciences division. These include our cell biology, molecular biology, protein and cell analysis, and sample prep segments. Each of these segments have individual reagents, instruments, and complete solutions for life sciences and diagnostics. I will be focusing today on the use of products and capabilities within our protein and cell analysis and sample prep segments, and how our customers can accelerate their immunodiagnostic product development and commercialization. With the emergence of COVID-19, our customers' product development and commercialization of diagnostics tests and vaccines has required the utmost urgency to provide various components required, along with the ability to meet the rapidly increasing scale required at the best quality. When our customers partner with Thermo Fisher, they have access to a complete portfolio of products and capabilities that enable faster assay development along with security of supply. As an example of how this works for our partners, I would like to start by reviewing some of our SARS-CoV-2 assay reagents. Thermo Fisher is one of the leading experts for development and manufacturing of immunoassays on various platforms. This slide shows our SARS-CoV-2 ELISA. We have used our years of expertise to develop some of the best serology assays to be able to measure SARS-CoV-2 specific antibodies in serum or plasma. Various research use applications can include things such as epidemiology studies, QC validation for vaccine development, or basic understanding of research of the virus. The combination of the spike trimer structure and the total antibody detection provides an accurate reading of SARS-CoV-2 antibody detection, thereby minimizing false positives or negatives. Assays can be run either qualitatively as a positive negative or as a quantitative assay using a standard curve. So Thermo Fisher products like this ELISA are available for internal research use only. So if a customer would like to use either this ELISA or perhaps one of the components within this ELISA in their products, they need to work with our global OEM team to confirm that we have the rights to offer this product for the customer's intended use. So that's what I want to highlight today is how do you take an existing product from Thermo Fisher and commercialize it in your diagnostic development. One of the key components of an ELISA are antibodies. Thermo Fisher offers a wide range of over 100,000 antibodies, and these include things such as these recombinant SARS-CoV-2 antibodies. All antibodies that customers would like to potentially use commercially should first be reviewed by Thermo Fisher to make sure that we're able to convey the commercial rights for the customer's intended use. Confirming commercial rights is just one way that our global OEM team supports our customers, and I will discuss how we do that over the next few slides. In addition to confirming commercial rights, our global OEM team offers our partners a variety of benefits. These include the ability to customize any of our products and services, access to our team of technical experts and resources to be able to scale along with our partners' commercialization, a long-term commitment that provides our partners with security of supply and business terms, and regulatory support to deliver the product specifications that are required by our partners, as well as change notification if something has to change. Our antibodies are provided for internal research use only. It is important to contact your global OEM representative to perform antibody review to make sure that you are able to commercialize your product for your intended use. Thermo Fisher sources our antibodies many different ways. So we have our own internal in-house antibodies, but we also in-license antibodies from third parties or are supplied from third parties. Depending on the antibody, some uses in our partners' products may not be permitted. To minimize risk for both us and our partners, Thermo Fisher has created a team of experts that review all intended uses of the antibodies that we offer through our global OEM channel. This is referred to as our antibody review team. It's also internally called ART, where we will obtain the necessary approvals where possible and execute supply and license agreements to ensure contractual compliance and thereby protect both our partners and Thermo Fisher. We offer this support to avoid potential issues in the future where an antibody is commercially used for an application that is not approved. There's nothing worse than for one of our partners to be procuring one of our antibodies and to find out that they could not use it for their commercial use at a later point. So we offer this review to make sure that up front we are giving the appropriate rights to our customers where possible. Thermo Fisher customers have access to the highest quality products with a desired lot-to-lot consistency. Having this comprehensive offering of immunodiagnostic reagents gives our customers one-stop sourcing, which can be especially beneficial when other suppliers do not have reagents. This schematic here shows various components that are found within an immunoassay. From the solid phase, which could be, for example, a magnetic bead or a latex particle, or even a ELISA plate, to the antibody that is conjugated to this bead or plate, as well as various buffers and detection reagents within the immunoassay. Thermo Fisher has the flexibility to manufacture each of these reagents as made to order, which helps maximize shelf life for our partners, as well as the ability to customize and scale with increasing demand, thereby minimizing the number of lots that are required. So this can be very important when our partners are looking to minimize costs, for example, for doing QC, but also for having more consistent results for their own products. Thermo Fisher does offer a wide variety of products for immunodiagnostics development. And on this slide, from left to right, you can see the 5 main categories of reagents and products that we offer, starting with our capture surface reagents. So this includes things such as avidin or streptavidin, nutravadin, as well as our latex particles that can be fluorescent, and our magnetic beads. The next category is our primary and secondary antibodies, which could be then labeled with various things such as dyes or enzymes, followed by our nonspecific binding category. So this represents things such as detergents and blocking buffers. The next category being conjugation. So this is where you're cross-linking various things or biotinylating various things in your immunoassay. And lastly, our substrates and dyes category, which includes some cutting-edge products within the chemiluminescent and fluorescent spaces. So this is substrates for alkaline phosphatase substrates and HRP substrates, as well as our dyes such as Alexaflor. So in terms of these categories, we make sure over our many years of supporting our partners in immunodiagnostics that these products deliver the quality and performance that our customers require. And our partners expect to be able to lean on us to provide the reproducibility and quality of these products. And as you look at this slide here, you can see that this is represented in different ways. So in terms of our capture surface reagents, for example, we really pride ourselves on being able to deliver the same performance from lot to lot with the same quality, so that our customers are not worried about that. And for any of our products, we want to be able to provide that reproducibility that is needed in immunodiagnostics. Once a product is developed by one of our partners, they need to understand what kind of tolerances that they have, and they rely on us to supply the same material over and over again. And if something happens, to be able to identify the problem quickly and resolve it. This is where open communication and partnering can be very important, and provide consistent products to our customers. So how to benefit from being our partner? We assist our partners by providing access to a team of experts that can help overcome issues when they arise for any of our immunodiagnostic products. The products can be customized to optimize performance. As a partner is developing their product, customization can be very valuable because it would help optimize performance. And performance can not just mean the performance of the assay, but things such as stability can be very important to our partners. So having the ability to customize and to scale can be a really helpful for our partners in terms of delivering the highest quality and a comprehensive partnership between us and them. And this includes things such as, the ability to confirm commercial rights, but also, change notification as well. There's nothing more concerning to not have any visibility to things that happen unbeknownst to our partners. So when a raw material supply is changed, we have the ability to confirm with our partners that has happened and help validate if necessary. So with that, I would like to highlight some of our manufacturing capabilities within Thermo Fisher. We have a team of synthetic organic chemists, both from an R&D and manufacturing perspective, that supports the production of a variety of different products. And these include things such as cross-linkers, biotinylation reagents, detection substrates. And by having this team, we are able to go from milligram to kilogram scale of these products, as well as the ability to customize things such as various quality control assays to meet the customer's needs. So this is a valuable resource for our partners to have access to this team of synthetic organic chemists. Similarly, we have a team of folks that specialize in the formulation of buffers and reagents and chromatographic supports. So what this means is, for a product that's a formulation, we can make lot sizes up to 5,000 liters in batch size, which when scaling is very important, the ability to not just make the 1 liter batch, but also to make up to 5,000 liter batches. And this goes across many different products, such as our blocking buffers, our detergents, our enzyme substrates. So having the ability to formulate and to make this scale, from milliliters to kiloliters, can be very important for our diagnostic partners. In terms of other capabilities we have in the areas of proteins, antibodies, and labeling, we offer various types of protein purification, bioconjugation, use of avidins and plate coating. We have abilities to lyophilization. So all of these protein and antibody modifications are available. These capabilities are available to our partners as they're developing their immunoassays. So these components can be very valuable to be able to support our partners with their internal development needs. We also have the abilities to package and finish products to our customers' requirements. So just because we offer a particular product in a specific bottle or packaging doesn't mean that that's the optimal packaging for our partners. So we have the ability to bottle all of our products in various ways. Could be into an ampule or a different size bottle. We can do powder pouching, dry box filling. We have automated systems for filling and capping. And we have the ability to kit a combination of our products if need be. So having these packaging options can help secure and deliver quality for our partners, and ability to take some of our capabilities and take them from our partners and do them internal for them so that they receive the material faster for their product development. Thermo Fisher has a team of quality control specialists that can perform a wide variety of tests to enable more consistent performance from lot to lot. By partnering with our team, we can customize these quality control tests in comparison to the products that are off the shelf. So we have our products that are on the web that are for sold for internal research use only, and they have specific quality control tests to make sure that they perform as intended. But sometimes for diagnostics, that's not enough. The range of the quality control tests, there should potentially be more of them, depending on the application, and it could be things such as smaller ranges of pH or other functional type of assays to help validate that the product that Thermo Fisher provides our partners is to the quality that they're expecting. So being able to customize quality control to any of our products is a very valuable resource to help with the commercialization process for our partners. Hand in hand with quality is our regulatory support. So our OEM partners require things such as lot-to-lot consistency of our components, and having the regulatory support for this is very valuable. So things such as change notification can be important. If something was to go wrong, the ability to audit our sites and work together to find what a potential issue might be and offer solutions is a valuable asset for our partners to be able to partner with us. And site audits are definitely an option for our partners to be able to understand what our processes are and how we could potentially improve things going forward. I believe it's important to understand the various centers of excellence that we have within Thermo Fisher, because it helps to have visibility to where these products and technologies and capabilities are coming from, and what they are at our various sites. So on this slide, we have listed many manufacturing sites within our protein and cell analysis business. Thermo Fisher is a combination of many different companies and technologies. Going from left to right here on this slide, would like to provide you a little bit of background on each site. So on the top left, our Eugene, Oregon site is our molecular probes business that many may be familiar with. This site specializes in things such as fluorescent dyes, including our Alexa Fluor and quantum dots, as well as latex particles and some conjugation technologies such as cyclic and instrumentation as well. So for example, our Attune flow cytometer is based there. Our Carlsbad, California site is a combination of our Invitrogen and eBioscience businesses as it relates to protein and cell analysis. This site specializes in flow, antibodies, and reagents. Our Rockford, Illinois site is our Pierce business, and they specialize in a wide range of proteins ranging from cross-linkers to detergents, blocking buffers, antibodies, and substrates. Next is the Bedford, Massachusetts site, which was our Tropics business, and they specialize in chemicals and substrates that are used in immunodiagnostics. Next is our Vienna, Austria site, which was our Bender business, and they focus on manufacturing of our ELISAs. Our Vilnius, Lithuania site performs protein production and is also a CGMP site for those partners that require a higher level of quality. All of these sites are ISO 9001, but the Vilnius site is, again, it's CGMP. And last on the list is our Bangalore site, and we do our antibody development in Bangalore. Developing an immunoassay is a partnership from the initial developments through the launch. Thermo Fisher's Global OEM and Licensing team supports our partners' development and commercialization of immunodiagnostic assays, letting them select a wide array of products and technologies. Our team of subject matter experts use their many years of experience to help guide and customize reagent selections and confirm that the commercial rights and necessary licenses are in place for the components that are being sourced. So working together with the Global team can help to ensure consistent performance and security of supply for important reagents. Post-launch, we work with our partners to maintain performance through features such as lot-to-lot consistency and change notification, and valuable mentorship and valuing our customers as well. So working together as a team, it's all about communication, ability to deliver products to our customers when they need it with the quality that they need. And with that, I'd like to thank you for staying on this webinar, and now we can answer any questions you may have.
Thank you, Anthony, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, where can I find more information on these Thermo Fisher products and capabilities?
Yeah, so this information is easily accessible on the web. I suggest you go to thermofisher.com/IDX, and that will take you directly to our OEM teams page, which has a lot of information on these products, as well as a nice tool called a flipbook, which goes into depth on many of these products. So you can download this flipbook, and it would take you to all the relevant web pages for these products.
Thank you. Next question. How long can it take to manufacture and ship an order, and what can be done to minimize the lead time?
Yeah, so this is a tough question because it can vary depending on the quantity and product that's being requested. So the range of lead time can be anywhere from 2 weeks to several months in terms of lead time, depending on what's being requested. So we have ways to help our customers and partners get product when they need it. So if it's something that's not forecasted and needs to be made to order, we suggest, working together with your global OEM partner to forecast potential needs so that we can stock things such as raw materials and intermediates to minimize these lead times. So that can take something that might be a several month lead time down to a couple of weeks. We do also have inventory, catalog inventory, that research use only inventory that we can utilize to help bridge any gaps in terms of waiting for our made to order products. You can potentially utilize some of our inventory that's in our warehouses to solve an urgent need.
Thank you. And it looks like we have time for one more question. Is there a team similar to the Goal team that works with customers for cell line licensing?
There is. It's actually part of the Goal team. There is a cell line licensing expert, and we can send you their contact information, and they would be happy to support you.
Well, thank you, Anthony. Do you have any final comments for our audience?
Just want to thank everyone for joining today's seminar, and please feel free to reach out to me if anyone has any questions on any of this content.
Well, thank you again, Anthony, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.